Hello, future nurses and science students. We are viruses, and we are the ultimate intracellular outlaws of the microscopic world. If you thought bacteria were simple, wait until you hear about us. We are so simple that scientists still argue over whether we are even technically alive. Unlike bacteria, we are not cells. We don't have a cell wall, cytoplasm, ribosomes, or homeostatic machinery, and we cannot generate our own energy, ATP. Because we lack the tools to reproduce on our own, we are classified as obligate intracellular parasites. This means we are completely helpless until we sneak inside one of your patient's healthy host cells, break into the control center, and violently hijack their cellular machinery to force them to build thousands of copies of us until they burst. The bare essentials: viral structural blueprint. While a human cell or a bacterium is packed with complex organelles, our structural blueprint is stripped down to the absolute bare minimum. We are essentially just a package of genetic material wrapped in a protein coat. 1. The genetic core, DNA or RNA. Inside a human cell, you always find double stranded DNA, but we like to mix things up. A virus can only possess one type of nucleic acid, either DNA or RNA, but never both. DNA viruses, highly stable. They sneak into the host cell's nucleus and copy themselves using the cell's normal replication tools. Examples: varicella zoster, chickenpox, hepatitis B. RNA viruses, highly mutable. They carry their own replication enzymes and multiply directly in the cytoplasm. Because RNA copying is messy and lacks error checking, these viruses mutate rapidly, which is why your patients need a brand new influenza vaccine or updated booster shots every single year. Examples: influenza, COVID-19. 2. The capsid, the protein armor. Our genetic core is surrounded and protected by a hard protein shell called a capsid. The capsid is made of interlocking protein blocks called capsomeres. It acts like a protective delivery capsule, shielding our fragile genes from being chewed up by the body's enzymes while we travel between host cells. 3. The envelope and spikes, the master keys, naked viruses. Some of us are naked, meaning the protein capsid is our outermost layer. Enveloped viruses. The sneakiest among us steal a wrapper from the host cell. As we burst out of a human cell, we wrap ourselves in a piece of that cell's own lipid membrane. The spikes, protruding out of our capsid or lipid envelope, are specialized protein keys called glycoprotein spikes. These spikes are engineered to mimic molecules the host cell naturally needs. They bind perfectly to specific receptors on the host cell's membrane, tricking the cell into opening its gates and swallowing us down. The life cycle, the 5 stages of viral replication. To understand how a viral infection develops in your patient, you must memorize the 5 sequential steps of the viral replication cycle. 1. Adsorption, attachment. The virus uses its glycoprotein spikes to lock onto a specific receptor on the surface of a vulnerable host cell. Nursing note: this specificity determines the virus's tissue tropism. For example, the influenza virus binds specifically to respiratory epithelial cells, while HIV binds exclusively to the CD4 plus receptor on Tommy the helper T cell. 2. Penetration and uncoating, the break-in. The host cell swallows the virus via endocytosis, or the viral envelope fuses with the cell membrane. Once inside, the protein capsid dissolves, uncoating, releasing the viral DNA or RNA free into the cell's cytoplasm. 3. Synthesis, the hijack. The viral genetic material takes total control of the cell. It shuts down the host cell's normal protein synthesis and commands the cell's ribosomes and enzymes to work overtime, manufacturing thousands of duplicate copies of viral genes and viral capsid proteins. 4. Assembly, the factory line. The newly manufactured viral parts spontaneously snap together like an automated assembly line, packaging the fresh genetic cores inside the brand new protein capsids. 5. Release, the burst. The host cell becomes packed to maximum capacity with thousands of mature, identical viruses. The viruses release enzymes that tear the cell membrane open, violently lysing and destroying the host cell as they spill out into the surrounding tissue to invade neighboring healthy cells. Retroviruses, flipping the script. As a nursing student, you will deal extensively with a highly specialized subcategory of RNA viruses called retroviruses, with the primary culprit being HIV. Normally, transcription goes from DNA to RNA, but retroviruses carry a unique specialized enzyme called reverse transcriptase. When they inject their RNA into a host cell, this enzyme converts their viral RNA backward into viral DNA. They then use another enzyme called integrase to stitch that newly made viral DNA directly into the patient's own chromosomes. The virus becomes a permanent hidden piece of the patient's DNA, a provirus, allowing it to lie completely dormant for years before activating. Common viral conditions and pathologies. 1. Latent viral infections. Some DNA viruses don't kill the host cell immediately. After the initial infection clears, they crawl up sensory nerve pathways and go to sleep inside the nerve ganglia, entering a state of latency. The clinical manifestation. Decades later, when your patient experiences extreme stress, illness, or immunosuppression, the virus wakes up, travels back down the nerve pathway, and causes a painful, localized, blistering rash along a specific dermatome line. This is exactly how the childhood chickenpox virus reemerges later in life as shingles, herpes zoster. 2. Viral oncogenesis, cancer links. Certain viruses can alter the genetic structure of host cells so severely that they accidentally turn on cancer-causing genes, oncogenes, or disable tumor suppressor proteins. Critical examples: human papillomavirus, HPV, integrates into cervical cells, significantly increasing the risk of cervical cancer. Chronic hepatitis B and C viruses cause continuous inflammation that mutates liver hepatocytes, leading to liver cancer, hepatocellular carcinoma. Critical nursing implications and antiviral pharmacology. Antibiotics do not kill viruses. This is the number one piece of patient education you will provide in clinical practice. Antibiotics are designed specifically to target bacterial structures like peptidoglycan cell walls or bacterial ribosomes. Because viruses do not possess these components, antibiotics are 100% useless against viral infections like the common cold, acute bronchitis, or the flu. Misusing them only creates dangerous, resistant superbugs. The antiviral action. Because viruses hide completely inside your patient's own cells and use human machinery to replicate, creating drugs that kill the virus without accidentally killing the human host cells is incredibly difficult. Most antiviral medications, like acyclovir for herpes or oseltamivir, Tamiflu for influenza, are not virucidal. They don't physically destroy the virus. Instead, they are virostatic. They act as chain terminators that temporarily jam the viral synthesis or release stages to slow down replication, giving Tommy the T cell and Luke the lymphocyte time to catch up and clear the infection. Please like, comment, share, and follow for more.